Hello National Fives and welcome to this short little video explaining how we can measure respiration using apparatus in the lab. This little video is going to take you through a little recap on the process of respiration and the level of detail you need to know for National 5, and then the experimental techniques we can use to measure the rates of respiration and some of the features of that kind of setup that will help you tackle some questions at National 5. So, here is your recap. Remember, we are starting with a molecule of glucose, or some glucose, which is a sugar. We want to break down this glucose to release chemical energy that can then be used for our cells. So stage 1 of aerobic respiration, and that is respiration with oxygen, we are going to break down that glucose into a molecule that's called pyruvate. And that pyruvate is our kind of intermediate step of this process. By breaking down glucose into pyruvate, we release 2 ATP, which is our high energy molecule that can be then used to power functions in the cell. So we release 2 ATP, and all of this has happened in the cytoplasm. So that is stage 1. Stage 2 of your aerobic respiration, we are then breaking that pyruvate down further, and we are going to produce ATP, so energy. We're going to produce carbon dioxide and water. This stage 2 of aerobic respiration happens in the mitochondria, and it's the stage 2 that's going to release lots and lots of ATP for us. So it's the kind of the high energy stage, whereas stage 1 is only releasing 2 ATP. If we are carrying out respiration where there is no oxygen, we call that fermentation. And depending on whether we are looking at animal cells or plant and fungi cells, there are different pathways. So if we are in an animal cell, we are starting, stage 1 is before we start with glucose. We break that down into pyruvate and release 2 ATP. Because there is no oxygen, that pyruvate cannot be broken down further, so we break that down into a chemical that's called lactate instead. There is no energy release, so no ATP is produced in this breakdown. However, if the oxygen debt is repaid, so if oxygen then becomes available in this process, we can then convert that lactate back to pyruvate and go down normal aerobic respiration pathway to release lots of energy. So in your fermentation in animal cells, so in essentially our cells, we have glucose to pyruvate, pyruvate to lactate. If oxygen becomes available, that lactate can go back to pyruvate and then go on the normal pathway. In plant and fungi cells, it's a little bit different. Your glucose gets broken down into pyruvate as before, but this time, when there is no oxygen present, that pyruvate will be converted into carbon dioxide and ethanol, which is a type of alcohol. In fermentation in animal, plant and yeast cells, you will only ever gain 2 ATP from these processes. So 2 ATP will be released in stage 1 when the glucose is converted to pyruvate, but you will get no energy released after that, so it's only ever 2 ATP. Remember that respiration is controlled by enzymes. That means that the rate of respiration will be affected by temperature. If the temperature is far too hot, those enzymes will become denatured, or if the pH is extreme, the enzymes will become denatured. The enzymes won't work, which means they won't control respiration, so respiration would stop, because respiration is an enzyme-controlled process. That's a very quick summary recap of what you need to know so far. What we'll now look at is how we can measure respiration using different pieces of equipment. So, this apparatus that you can see in front of you is called a respirometer, and you will probably encounter these, or a variation of this, in kind of National 5 respiration questions. What this equipment is set up to do is to measure the rate of oxygen uptake by a living thing. So how much oxygen is getting taken up over time. Because if you remember back to our summary kind of word equation for respiration, if we're looking at aerobic respiration, we require oxygen. If there is a high uptake of oxygen, that would suggest that there is a fast rate of respiration. So we're looking at how fast oxygen is being used to give us an indication of how fast or how much respiration is happening. So if you look at the apparatus that's in front of us just now, you have got a beaker of coloured liquid, a ruler or some kind of measuring scale. This is like a little glass, we call it a capillary tube, but it's just a really narrow, thin glass tube, which is feeding into a boiling tube with a few different things. So these wee squiggly lines here are worms, caterpillars, something that's living. Okay, so we put a living organism, not humans, something small, into these respirometers to measure their respiration. So we've got something living here. They are placed on, so that dotted line there, imagine it's just like some mesh, so like a kind of metal mesh where there's holes where gas can pass through really easily. Below that, there's some beads. Now, it might tell you that they're sodium hydroxide beads. It could just tell you that it's a chemical bead. The point of these is to absorb carbon dioxide out of this experiment. Okay, so these chemicals that are in these beads, their purpose is to absorb carbon dioxide. At the top here, there is a syringe, and its function is to basically just seal off this experiment. So what you'll see is the capillary tube is feeding down into the liquid at the bottom here. There's no way any gas from the surroundings, so there's no atmospheric gases, can pass into this apparatus. Everything is sealed in this boiling tube and in the capillary tube. Okay, now let's think about what's happening with these gases. So the worms, let's just call them worms, they are respiring, so they're going to start to use up oxygen. So in that boiling tube, they're going to pull in and they're going to use up the oxygen that's available to them in this small space. As they carry out respiration and they're using that oxygen up and they're breaking down glucose and they're carrying out the process, they're going to release carbon dioxide. So they're going to breathe that out, essentially. That carbon dioxide, if it was in a normal situation, it would just remain in the atmosphere, but because we have got these pellets with chemicals, that carbon dioxide is going to be absorbed by that chemical. So it's going to be taken out of the air, essentially, in that beaker. So what we're going to essentially start to create, it's almost like a bit of a vacuum, because you're using up the oxygen from the air. The carbon dioxide that's released out is then being absorbed. So you're actually reducing the amount of gas that's in this kind of sealed experiment. So what you'll find is that the liquid that's in the bottom of the beaker here, it's going to start to rise up the side of, or it's going to rise up the tube next to the scale here. So you can start to see, as all the gas gets used up in this experiment, the liquid's going to get sucked up the tube. So it's like a straw. So you've made a bit of a vacuum and that liquid's getting raised up. What you can do is over time, so say over a period of 5 minutes, you can measure the height of this coloured liquid, which will give you an indication of the rate of oxygen uptake. So you could measure it over 5 minutes and see how much has been used up. You could then compare that to different other organisms. You could compare how much liquid is pulled up over a range of temperatures. You could compare a living versus non-living organism. It just depends on the design of your experiment. But the general idea here is that you've got something living that's carrying out respiration. It's using up the oxygen from its surroundings, but it's in a closed surroundings. As the oxygen is being used up, the organism will release carbon dioxide. That's absorbed by the pellets or the beads at the bottom of the boiling tube. That pulls the liquid up, and the rate, so how fast that liquid moves up the scale, is showing you how fast respiration is happening. The next piece of equipment we'll look at is how we could possibly measure fermentation. So we're still looking at respiration, but this is now going to be respiration without oxygen. So in the case here, we are looking at yeast cells and how we can get yeast cells to respire. So in the conical flask there, you can see you have got a solution of yeast that has been mixed in with some glucose solution. So that's, you've given the yeast some sugar that they can feed on. However, you'll see above your glucose and yeast suspension, you have got a layer of oil. Okay, thinking about the fact we're looking at fermentation, we don't want to have any oxygen in this experiment. So what the oil does over the top, it just floats on the top and it seals the experiment below from the air above in the conical flask. So you're going to have no oxygen in that surroundings. So it has to be fermentation what happens. If the oil wasn't there, it would be aerobic respiration because the yeast would use up the oxygen that's above them in the conical flask there. You will see that out of the conical flask we have got another little tube there that is feeding into a boiling tube where it says bubbles of carbon dioxide. So compared to last experiment with the respirometer, we were looking at how much oxygen is being used up. In this one here, we're looking at how much carbon dioxide is being given off. So we're looking here at the products of fermentation. So what we could do over time is count the number of bubbles that are given off over a set period of time. So say you set the timer for 1 minute, you count 20 bubbles, that's great. You could then change the temperature. So you can see this here is set up with a thermometer and a water bath. You could increase the temperature slightly, count the number of bubbles again, and see what effect temperature has on the fermentation of these yeast. So this version here, if we're looking at fermentation, you want to make sure no oxygen can get into your experiment. So we put a layer of oil over the top, and we're measuring the output of carbon dioxide, which gives us an idea of how fast that respiration is happening. So if we've got lots of bubbles, lots of respiration happening. If we've got no bubbles at all, that would suggest that there is no respiration happening. So these are two ways that you can measure respiration, and these are both diagrams that have come from SQA past papers. What you will find is as you do more questions, you'll see variations in the apparatus setup, just depending on what the experiment is out to prove. However, the key skill is that you can understand the basics of what gases are being used up, what gases are being given off, what does the rate or the speed that these gases are being used up or given off tell us about the rate of respiration. Why, for example, is a layer of oil used when we're investigating fermentation, and so on. So the key skill here is applying your respiration knowledge to the design of the apparatus. You might in the future be asked how you could improve an experiment, and so on. So this is very much an introduction. The thing you need to go and do now is go and practice questions like there is no tomorrow, and hopefully you will start to build this skill of improving experiments and evaluating what's going on in front of you on paper. So good luck and keep aerobically respiring. Bye!